Morning students, today you are going to study about the development of dicot embryo. It's a continuation of the fertilization. What is fertilization? You can recall what I have taught already. The fusion of male and female gamete. In angiosperm, the type of fertilization is double fertilization because the two male gametes are involved in fertilization. One male gamete fuses with the egg resulting in the zygote. Then another male gamete fuses with the polar nucleus and results in the primary endosperm nucleus. That primary endosperm nucleus only developing into endosperm. That endosperm is the nutritive tissue for the developing embryo. Okay, let's see the development of dicot embryo. The first cell is the zygote. The first cell is the zygote. So the embryo develops at the micropylar end of the embryo sac. The first cell, the zygote, it will undergo a transverse division and it will result in the two cells, the upper terminal cell and the lower basal cell. This is the two-celled conditions. Then the zygote, it will undergo a series of division and it will acquire a different shapes for the developing mature embryo. The first one is the globular, then second one is the heart-shaped embryo. At last, after crossing this globular shape and heart-shaped embryo, the embryo will develop into a mature embryo. The mature embryo consists of cotyledons, then radicle, it is covered by the root cap as well as the plumule. Next we can see what is seed. I have already taught you in the post-fertilization changes itself. The fertilized ovule only become a seed. Then it possesses an embryo, endosperm, and it has a protective coat. Here the seeds we can divide into two types, endospermous seeds. The seeds with endosperm we can call that as a endospermous seeds. Example, wheat, maize. Then non-endospermous, the seeds without endosperm we can call that as a non-endospermous. Example, bean and mango. Let's see the structure of the dicot seed. Structure of a scissor seed. It is an example of dicot seed. Here it consists of two cotyledons. Here the seed is covered by a seed coat. Outer testa, it is hard. Then inner tegmen, it is thin and membranous. Then the seeds are covered by a seed coat. Then it is attached to the fruit wall that is called funiculus, and it is leaving a scar that is called hilum. Then the seed consists of a small pore that is called micropyle. It is helping for exchange of the gases as well as water entry during germination. Which one? Micropyle. Then here it consists of two cotyledon. Then the plumule, it is the upper axis. The two cotyledons bearing upper axis and lower axis. The upper axis is the plumule. It is otherwise called embryonic shoot system. Then the lower axis, it is the radicle. It is the embryonic root. The plumule will develop into shoot system. Then radicle will develop into root system. Yes. Then what is epicotyle? Here epi, above, cotyle, cotyledon. So the embryonal axis above the cotyledon, this part, the embryonal axis above the cotyledon, that is called epicotyle. It will ends in the plumule. Then hypo, below, otherwise at the level. So the embryonal axis, this part, the embryonal axis below or at the level of cotyledon, that is called hypocotyle. It will ends in the radical part. So scissor seed, it consists of two cotyledons. Then the two cotyledons will be attached in the embryonal axis. It stores food material in the pea plant. Here seed coat, outer testa, inner tegmen. Then the plumule, embryonic shoot. Then radicle, embryonic shoot. Next we can see structure of oryza. Have you all eaten rice? Yes, here the oryza is an example of monocot seed. Monocot seed because it consists of only one cotyledon. The fruit name is called caryopsis. It's a one-seeded fruit. So we can call this as a caryopsis. What is that? Caryopsis. Here each seed remains enclosed by a brownish husk that consists of a glooms that is arranged in two rows. Then the seed coat is a brownish colored layer that is adhered to the grain. Then the endosperm, it is a storage tissue. Then it is separated from the cotyledon by a epithelium. This layer is called epithelium. Here the scutellum, one shield shaped cotyledon will be there. That name is called scutellum. What is the shape of the cotyledon? Shield shape. Then here it will be at the lateral side. Then it consists of the shoot apex, it is the plumule. Then here it is the radicle. So radicle is enclosed by the root cap. The peculiar character of monocot seed is the radicle is covered by a sheath that is called coleorhiza. Then the plumule is covered by a sheath that is called coleoptile. This is the peculiar character of the monocot seed. Here the scutellum. This is the scutellum is the which one? Cotyledon. Only one shield shaped cotyledon, that is name is scutellum. It supplies the growing embryo with the food material from the endosperm absorbed by the epithelium. So the scutellum supplying the food for the developing embryo from the endosperm by the epithelium. So structure of oryza seed, monocot seed. How many cotyledon? Only one cotyledon. What is the shape? One shield shape. What is the name? Scutellum. What is the name? Scutellum. What is the peculiar character? Yes, radicle is covered by a sheath, coleorhiza. Then the plumule is covered by a sheath, coleoptile. Next we can see apomixis. Apomixis, it's a type of reproduction, does not involve union of male and female gamete. Normally, sexual reproduction means which involve the fusion of male and female gamete that result in the zygote. Here, it does not involve union of male and female gamete. This is apomixis. The term apomixis was given by the scientist Winkler in the year 1908. So here they have given definition. Apomixis is the substitution of the usual sexual system which does not involve meiosis and syngamy. So here it's a type of reproduction which does not involve fusion of male and female gamete. This is apomixis. So the fusion of male and female gamete, that type of reproduction is amphimixis. Here apomixis are of two types, recurrent and non-recurrent. Here recurrent includes vegetative reproduction and agamospermy. Then non-recurrent means, you know very well, the haploid embryo sac will be developed after meiosis. That haploid embryo sac will develop into an embryo without fertilization. Normally after fertilization only embryo will develop. But here the haploid embryo sac will develop into a new embryo. That is non-recurrent. What is that? Non-recurrent. Here recurrent, first vegetative reproduction. Here the any part of the plant may develop into a new plant. That is called vegetative reproduction. Next, agamospermy. Agamospermy means embryos are formed without meiosis and syngamy. No gamete formation, no fusion of male and female gamete. That is agamospermy. There are three types. First one adventive embryonic, second one diplospory, then the third one apospory. Let's see one by one. Here first one adventive embryony. Here the embryos are directly formed from the diploid sporophytic cells. It may either from nucellus or it may from the integument. So it is called sporophytic budding. So it may develop from the nucellus or integument. So we can call this as a sporophytic budding, because the gametophytic phase is absent. Example, citrus. Next one, diplospory. Here a diploid embryo sac is formed from the megaspore mother cells without meiosis. So diploid embryo sac will be developed from megaspore mother cell without meiosis, that is diplospory. Example, ayarva. Then the last one, apospory. Here you know very well, megaspore mother cell undergo meiotic division. It results in the 4 megaspores. Any one will be developed into embryo sac. But here all 4 megaspores will disappear The new cellular cell become activated and develop into diploid embryo sac. This is apospory. Otherwise, we can call as a somatic apospory. Somatic apospory, because the body cells, new cellular cells only get activated. So, we are giving a name somatic apospory. Example, Parthenium. Next, we can see the polyembryony. Polyembryony, it's a occurrence of more than one embryo in a seed. I mean, the formation of more than one embryo in a seed is called polyembryony. Here, the first case was reported in oranges by the scientist Anton van Leeuwenhoek in the year of 1719. So, usually only one embryo will be there in a seed, but there is a chance of many embryo in a seed. That condition is called polyembryony. There are four categories of polyembryony based on its origin. First one, cleavage polyembryony. I mean, the cleavage mean division. So that type is found in the orchids. Next one, formation of embryo by cells of embryo sacs other than the egg. Usually the fusion of egg and the male gamete results in the embryo. But here other than the egg, synergids, example Aristolochia, then antipodal cells may develop into embryo in Almus, then endosperm may develop into embryo in the Balanophora. Then development of more than one embryo sac within the same ovule. Only one embryo sac in the ovule, but here development of more than one embryo sac within the same ovule. Then the last one, activation of the some sporophytic cells of the ovule. Have you remember about apospory? Yeah, that same time. So here that megaspore cells may degenerate. So the new cellus part, otherwise integument part, may develop into embryo. The new cellus part citrus, then integument part may develop into embryo in syzygium. Then what is the usage of the polyembryony? So here it will be help for producing virus free plants. Then it is better clone for orchids. It's a better clones for orchids. So it is commercially very useful. Next we can see parthenocarpy. Next we can see parthenocarpy. You know very well parthenocarpy. The fruits are formed without fertilization. Usually the fruits are formed after fertilization. Here the fruits are formed without the act of fertilization, so definitely no seeds will be formed. Seedless fruits will be formed, so it is very commercially useful. Banana, grapes and papaya. Then NITS scientist in the year 1963 classified three types. First one, genetic parthenocarpy. Second one, environmental parthenocarpy. Then the third one, chemically induced parthenocarpy. First one, genetic parthenocarpy. Here it is due to hybridization, otherwise mutation. Hybridization, otherwise mutation. Some sudden change. Otherwise, if we want to get a desired character, so we are crossing the plants. In that type, we can produce this genetic parthenocarpy. Example, citrus, cucurbita. Then environmental parthenocarpy. Some environmental conditions, it may be frost, fog, low temperature also induces parthenocarpy. For example, low temperature for 3 to 19 hours induces parthenocarpy in pear fruits. So environment also induce parthenocarpy. Then chemically induced parthenocarpy. See applications of some growth promoting substance, plant hormones, that auxins and gibberellin induce parthenocarpy. So here mutation, otherwise hybridization also induce parthenocarpy, then environmental condition as well as the plant hormones. Let's see what are the significance. Here seedless fruits have great significance in the horticulture department, then great commercial importance. Then it is used for preparing jam, jellies, etc. Then here the high proportion of edible part is available because the seed is not found. So it is very commercially useful in the horticulture department, and many other products forming also, the jam, jelly. Dear students, I'll give some 5-mark question. You can study this and write as a self-test. I hope you have understood this lesson very well. What are the questions I have given as a self-test? You can study thoroughly. Thank you.